session before the break was in some ways kind of a little unsettling. We talked about airline near misses and security problems with our smartphones and so on. So we thought we'd lighten the mood a little bit by now talking about cancer. Okay, I apologize for that. And to help us out with this, I'm delighted to be joined by Shirley Pepke and Greg Versteeg. Shirley's a computational biologist and now works for Lyrid LLC. And Greg is a research professor at USC's Information Sciences Institute. They're choreographed so that Shirley's going to be on this side, Greg's over there. So please join me in welcoming Shirley and Greg.
So this is basically our story of how we tackled the real-world problem of cancer, which was a very personal problem for me, and used big data in the context of our collaboration to hopefully, apparently, to change the outcome in my case. And so we'll just go ahead and get started. So this is a picture of me and my family in September 8th, 2013, my son and daughter and my husband. And we're all pretty happy. We all look pretty healthy. But then just 2 weeks later, it turns out I was diagnosed with ovarian cancer. And ovarian cancer is typically diagnosed at a very advanced stage, as mine was. It was all over the abdomen. They even had to scrape it off my diaphragm and my abdominal wall. So at stage 3C, ovarian cancer has a 5-year survival of only 36%. So you can imagine, so my son is 9, my daughter's 3. This is a very unhappy event and totally unexpected. And so there's a certain amount of shock that comes with that diagnosis. But then the next question is, well, what do you do? And some people just sit back and say, well, I'm going to do whatever my doctors tell me and hope that the roulette wheel goes in my favor. But that was not me, because I had a background in a lot of different things related to data analysis. So I had my Ph.D. in physics, but then had moved in almost immediately into engineering and computational biology, and had been doing that for many years. And in particular, in the context of both physics and genomics and even AI and neurobiology, I had done almost either all data analysis or model fitting. So I had this background to understand the biology of what was going on. And so it seemed just an obligation at this point. Well, this is a problem for me, right? It's a biology problem, and it's a problem that I can work on. So that's what I was going to do. But I couldn't do it on my own, obviously. So the first thing I would need was my tumor data. And in particular, I would need sequence data. And this was not just mutational data, which is what most people use in the clinic. And to this day, that's exclusively what's used in the clinic for the most part. But it was also gene expression data. And not just gene expression and mutation data for a few genes, but I wanted it for the whole set of genes in my entire tumor. So this is not something that patients commonly have access to. So I consulted with some colleagues and got directed to the Translational Genomics Institute, headed by Jeff Trent. And they put me under research experimental trial to get my data returned to me. So I could, in effect, do whatever I wanted with my data, including analyze it myself, which was a scary proposition, but the other alternatives were worse. So as I said, it was a lot of data that isn't typically available. But then, in addition to the data, I needed to figure out how to understand it. Because this data was not commonly used in the clinic, the problem was that no one knew how to interpret it for clinical purposes. And I knew that in my case it would be important, because ovarian cancer in particular doesn't have a lot of genetic mutations. So your question is, well, what's going on? Well, we're not sure. There are several possibilities, but one of the possibilities is that there's an alteration of gene copy numbers. And the copy number of the gene results in a different amount of protein being made. And so it's not something you would see by looking for alterations of the DNA sequence, but you would see it in gene expression data. So this is why I decided, well, this is what I have to look at if I'm going to make some progress. So I needed to put that in the context of other tumors to try and understand, well, how does this impact, what does this mean for tumor biology, and how does it impact outcome? And so, I was about to undergo chemotherapy, and I knew I needed some heavy-duty algorithmic analysis to do that. So I looked for help in that direction too. And I consulted with my friend, good friend, Christina Lehrman, who happens to be a research professor in computer science at USC. And she said, oh, I know some people who are doing machine learning and are interested in that. And she connected me to Greg for Steeg. And so I looked at what Greg was doing, and it seemed like he was developing algorithms that were exactly of the type that I thought would be helpful in looking at the gene expression data. I'll let Greg tell you a little bit about what he was thinking about before we started this project in terms of machine learning.
Okay. Hey, oh yeah, my mic is on. Great. Hi, I'm Greg. So like Shirley, my background is also in physics. The things I was drawn to were all like kind of things that crackpots are drawn to, like crazy things. So not immediately really relevant for what we're talking about today. But there is one thing that kind of strikes me as compelling about what ties these together. And in these cases, the tool that really helps us understand what's going on in those complex systems is information theory. It sort of gives us the right way to break things down and understand what's happening. So even though I found that very exciting and compelling, I also got like really disillusioned working on this types of stuff. It just feels like, you know, even if you do something really great, like the real world impact is kind of, in the immediate impact is really limited. So I thought, right, so this is useless stuff. I want to do something with some real world impact. So maybe I should apply these tools, which are so great, to a more useful field like biology. So that would be great. There's only one tiny problem, which is I don't actually know a single thing about biology. But that's never deterred any physicists from doing this type of thing. So I went in this direction. And
I about cancer biology.
And Shirley asked me last night, we were looking at the slides, she's like, did you really think you wouldn't have to learn any biology? I was like, yeah, I really thought that. So that was my, so, you know, for my approach to the problem, I thought, oh, like, how can we learn about biology without knowing anything about biology? So I think about this like a learning problem, and I want to kind of give you a sense of like how I was thinking about this problem, and also to kind of distinguish it from what I think are the most popular machine learning frameworks that most people think about. So if you imagine that your data is something really cute, like these kittens, or like these images, the normal, like the most successful and kind of widely publicized types of machine learning algorithms you hear about are on this side. They're supervised learning algorithms. And when you use supervised learning, you actually have to know just tons of stuff at the beginning. So first of all, you have to know what you're looking for, which is a big deal, right? And you say, okay, well, I know what I'm looking for. I'm looking for cats. Now, not only do you have to know what you're looking for, you have to already have a way to recognize and label some things that are cats and are not cats. So you have to do a lot of work to get this. And at the end of the day, you're going to get train a black box prediction algorithm, which is an amazing feat that this has come so far. And this algorithm is going to tell you, you'll give it a new image, and it'll tell you whether you've got a cat or not. So that's amazing, but you're only going to find cats, first of all, and you had to know a lot about cats before you could even, you know, get into this, the ballpark of something useful here. So, you know, for contrast, the type of learning, you know, if I want to take this position of like total ignorance, where I have to do something more like this unsupervised column. So in this case, we don't know what we're looking for. So you just kind of have this idea of like, yeah, what's interesting in the data, which is a pretty difficult place to start. So what you'd really like is something like to say, oh, you know, there are some hidden factors in this data, and these two pictures have this hidden factor, and this one doesn't. So it'd be cool if you had an algorithm that could tell you something like that, right? Without you telling what you're looking for. And you know, in this case, you might say, okay, so additionally, maybe the algorithm could tell you something like, you know, this hidden factor which exists in these two pictures but not in this picture has to do with pixels over here and over here, right? So that's getting in the ballpark. It told you something you didn't know, told you something you weren't looking for, and there's one more challenge, though. Maybe it could give you all that information, but what we really want is not, here are some pixels that are informative about this hidden factor. What we really want to know about is hats, right? And this is like a big leap still. So this was how I was coming at the learning problem, and you know, so this was around the time Shirley and I met, and now we're going to kind of compare to, you know, what it is Shirley was really trying to do with her data.
Right. So coming into this meeting with Greg, so I thought about, well, what do I need to do with in terms of the ovarian cancer analysis? Well, for one thing, I wanted to take this tumor data. So if you see on the right, there's a picture, and on the vertical axis is genes, discrete genes, and on the horizontal axis are different patients or tumors, actually tumor samples. And so green is low expression and red is high expression. So you can see that there's a spectrum, there's an ordering of these tumors with respect to these patterns of gene expression. And so what most people at the time were doing in terms of looking at ovarian cancer tumor analysis is they would tend to categorize patients as one of 3 or 4 subtypes. Well, and these were mutually exclusive subtypes. You're either type A or type B or type C or type D. Problem, what if your tumor doesn't clearly align with the subtype, which was fairly common, as you might imagine, when you have this sort of behavior. And it turns out mine didn't. Mine was a more complicated tumor. So that was one of my goals, was to actually, instead of looking at these subtypes, these mutually exclusive subtypes, to just look at activated networks in the tumors that were biologically meaningful. So networks being anything up to maybe dozens of genes that are interacting to affect some function in the tumor. So I was also thinking about the fact that whatever patterns were learned should be learned in a hierarchical context. For instance, if you find a network associated with inflammation in the body and a network associated with white blood cell movement, then if you would have a hierarchy, then up above those you would have an immune response network that would be basically the aggregation of both of those. And so the reason I thought that was important was because this was exactly the way biology was organized. So I thought this is how you get to interpretability. You have to be as close to the domain as possible in terms of its organization to understand what the outcomes mean in terms of clinical results. So also the other thing I was thinking about was the fact that the genes shouldn't be in one group or another, because nature loves to reuse things. Genes are always involved in multiple networks. So especially if you're looking for very small networks of a few, maybe a dozen or 2 dozen genes, then if you say that they can only be in one or the other, then you're just reducing your signal level in order that you can use to find that functional grouping. And so that was important, and that was something that not a lot of algorithms that I was familiar with or that were being used could do. And again, it played into interpretability for the data. So Greg has these ideas, and I have my own ideas about this domain, and we decide to, I contact him, and we meet up at a cafe because we were not at the same institution. And so I told him about the data, my problem. So you see, I'm thinking about the networks and what I need to get out of the data, the gene expression data. And Greg is nodding politely. And then he starts to tell me about what he's been thinking about in terms of a learning algorithm. And I'm in turn nodding politely. But we seem to have enough of a shared set of goals that actually, from that first meeting, we were able to get a set of concrete tasks and actually agree, well, this is what we're going to do. We thought about the matrix and what it would contain, the training matrix, and said, okay, and where the data might come from, and what the learning algorithm would do. And we actually went off and decided to get back in touch when we had some of these things. But although we had some shared goals and some things that were promising, there were also a lot of big challenges that needed to be addressed. And so on both sides, on both the machine learning and the domain-specific side, domain interpretability side. So first, Greg's going to tell you about the challenges of the machine learning.
My favorite challenge. So this is one that's kind of a generic challenge, and I think maybe would affect a lot of people here thinking about big data. So a lot of the talks about big data is like, oh, big data is good, right? It can only be good. The more data, the better. So a lot of data that we heard about, like with malware, it was like, oh yeah, give as many samples as you can, right? You're always doing better if you can get more samples of your data. But some big data has a different form. If you rotate this type of data, it's no longer called big data. So if instead you have many variables of data, but only a few samples, that's actually bad news, and it has another name, and that's the curse of dimensionality. And there's a really strong reason why we call this a curse, because a lot of algorithms, their computational running time depends on the dimension. If your dimensionality goes up, you need at least as many samples, or maybe even quadratically more samples, or some crazy requirements like this. So a lot of times, if your data is growing in this way but not in this way, there's a lot of stuff you can't do. You're going to have a lot of problems. So the reason I bring this up is that this was one of the main challenges we had to solve, because gene expression is cursed in exactly this way. When you look at gene expression data, you'll get tens of thousands of genes quite easily. And if you look at a single clinical study, it's expensive to do a clinical study. You can only get maybe a few hundred people, and actually even more than that, right?
Right. In ovarian cancer, I mean, there are 20,000 cases diagnosed a year, so you're never going to get more than, 1,000 or 2 samples in a study at best.
Yeah. So even if you could collect data from every single ovarian cancer case, it still wouldn't really qualify as big data in the way that the things we've heard about. So that's a big challenge, and this is something I'll talk about a little later, is how we address this challenge. But now Shirley has some of the more domain-specific challenges.
Right. So at the same time, so I was looking at, so if we get this, so what do we want the algorithm to do, and how do we know that it's working, right? And so again, coming at this as a person who's not a cancer biologist, I needed the algorithm to teach me about the biology, but I couldn't start from nothing. So I had to address these questions of how are we going to interpret biological information in the hierarchy in a systematic way? How are we going to judge what matters for clinical outcomes? And eventually, we went with survival analysis, which is common. But most importantly, how to use those outcomes associations to choose treatments and combinations of treatments, right? Which is where I wanted to go with it. And then how to measure the robustness of the results in a way that aligns with the interpretability. So you want to be sure that anything you get is reproducible enough so that you feel good about giving a patient a drug based upon it, right? And one way to get to that robustness is to look at the biology that underlies the groups that you find, right? And to look at how that's impacted by different variations in the algorithm or the data. And so basically, there were more challenges, but basically the problem came down to how do we translate the numbers and the algorithm output to lifting the survival curve. So this is what's commonly looked at in terms of cancer. On the y-axis, you have the fraction surviving. On the horizontal axis, you have months survival, and this is actually for the Cancer Genome Atlas cohort for ovarian cancer patients, and you can see the median is less than is just about 44 months, and so it seems very. So the goal is to actually raise that survival curve so that, in general, given a drug, someone, and especially me, would survive longer. So the next year and a half, we went through this process of iteration. So Greg continued to develop CoreX, and I started working on the biological interpretability of it. And also I was working on organizing the training data. And all of this changed over time. So I got one set of data, I passed and pre-processed it, sent it on to Greg, who ran an early version of CoreX. Although, bear in mind that actually CoreX didn't officially exist at this time. There wasn't even a manuscript. So he's developing this, and he would feed some numbers back to me about these gene groups, and then I would look at the biology interpretation according to the tools I was using. And then, we would go around this way, and the data changed. At one point, we thought about using a particular type of technical gene expression data, and then I shifted to a different type because it turned out there was more better signal to noise ratio. And Greg kept developing CoreX, shifting the particulars of how that operated with the data. And then I kept developing the biological interpretation tools to make them more refined. And so we performed our roles, but it was mostly back and forth, right? So one person would do something and pass it back, like sort of a football. Okay, so what do you make of this? And then we'd have a discussion about it. And maybe the other person would go off and do something different, and then it would come back, and then we'd do the same thing. So, for most of that year and a half, we're both learning about the problem we're dealing with, essentially, right? And so I go through my initial chemotherapy, and I go into remission, and we're still working on this, and we have no idea whether it's really going to yield anything useful, because at first it doesn't. It's not close to what it needs to be. But we keep going, because, at the back of my mind, I'm thinking, this could determine when my life ends. And so, and Greg was, I'm sure, aware of that as well. And so the thing is, it wasn't a sort of a continuously progressive process. I think probably there were times when months went by where nothing really happened, and maybe we were working on other things or thinking about it. And then we would, someone would come up with something and say, oh, look at this. And then suddenly things would progress really quickly. So this is kind of how it was. We were kind of going on this roller coaster. And I think because we weren't at the same institution, there was kind of a benefit to the distance, because we didn't feel a pressure to really be, producing something all the time. And because we were both aware of what this was really about, which was this real-life problem of dealing with cancer, and in my particular case, especially, I think our motivations were different, and we persisted. So I think Greg has said that he felt at certain times there would have been it would have been easy to publish something at some conference based upon the results, but they weren't good enough to pick a treatment, so we just kept going. And so, and we kept going in some ways that were unconventional for the application. And so he's going to tell you about some of the things that he did with CoreX to make it work for the data during that time.
Yeah, so I do just want to give you, a quick sense of like, what was the idea behind this. So, I said I really wanted to start with this idea that we don't really know anything about the data. So the variables here are genes, and I'll be almost no math. So the idea is that you'd say, okay, what we really want to do is look for some hidden factors, which I call these Ys. The Ys are going to be some function of the genes, and we want a way to find something that's possibly useful, and I said I wanted to use information theory to do that. So the idea is really this. It's to say we're really looking for these hidden factors that maximize some information measure. And actually, there's a lot of ideas about how to use information theory in this way. And the thing that really sets this approach apart is that what we're really maximizing is not the information that Y has about the data, because that's really just memorizing data. We don't want to memorize the data. What we're maximizing is the information that Y has about the relationships in the data. So that's using a kind of multivariate mutual information to do that. So find Ys that tell you about relationships. So that's the idea behind the approach. And I want to just like tie back quickly to some of the things we've talked about. I said, this is really an unsupervised approach. So we don't know what it is we're looking for. We don't put labels in. But there really is something you have to know ahead of time, which is, we're saying we're looking for relationships in the data. And we really hope that in the gene expression data, those relationships are going to be useful. And with Shirley's help, we could see that was really true. So I mentioned the curse of dimensionality, and there's a few ways that plays in here, which I'll mostly skip. So one, this information theoretic measure is very robust to going into high dimensions because more variables leads to more relationships, which in a way helps the method. And we had a lot of iterations about how to kind of mitigate noise in the data that were kind of specific to this case. But I'll skip that. And one thing is that we get a lot out of this type of analysis. So we get these hierarchies of relationships between genes, and for each gene we get a hidden factor which explains the relationship in the genes it's connected to. And I put explain in scare quotes because it's a way that statistics people sometimes use the word explain. Which is far from what a human explanation is. So it really, it really means that once you've conditioned on this factor Y, then the variables will be conditionally independent. So that explained it, but it doesn't really explain it from the point of view of what we want. So what we get out of this is we get all the hierarchies and all these factors, and and there's really hundreds of the factors. So now we have hundreds of factors, and the question is, what are we going to do with all that? And really, without someone like Shirley, who could really understand the math at a deep level and really understood the biology at the right level, I don't think we could have done very much. So Shirley is going to give like a little bit of a sketch of, what it is exactly that we could draw from all this.
Well, basically, this is basically how it all came together. So one of the things I did was look at different database annotations that would associate with each of the factors. So I could run a bunch of scripts and get multiple types of functional information about the genes, such as what type of cellular processes they were associated with, what pathways they were associated with. And so when CoreX output, so this plot is a plot of the factors that CoreX found in one of the later runs. And so each node here is a grouping. And so most of the nodes are what we call the level 0 groups. But then when they come together, so that's a higher level in the hierarchy, right? That's the level 1 group. And so it turns out that when all the stars align and all the parameters are correct and everything has been developed, then, and Greg gave me this tree output from CoreX, and I looked at the annotations that were biological annotations. It turned out that CoreX was very nicely finding, within these 200 groupings, not only was it finding interesting biological function in the vast majority, which was very different from what other people had been able to do. So they would pull out maybe 20 groups out of this data. We were pulling out over 100 that looked like they were functionally significant. But it also could cluster them on the higher levels. So you could see that, well, most of the tumors were perturbed in terms of their immune system activation, But there were also lots of smaller groups that had perturbations in their gene expression associated with the tumor microenvironment or cellular replication, or even there are some groups associated with nerve growth, which I thought was interesting. At first I thought, well, is that wrong? But it turns out that there is associations of, there are tumors that exploit nerve growth factors to continue growing. And so that's a real thing. And so CoreX was teaching us about tumor biology. So this was published in BMC Medical Genomics in March of 2017. But in addition to the interpretation of the biology, so it turns out that there were also strong correlations with survival that associated with some of the biological groupings. So here I show one small network that was found by CoreX that was associated with metastasis and invasion, so it indicates very aggressive cancer, and another small network that was found that was associated with a good immune activation. It turns out if your immune system is activated, it can clear the cancer better when you have chemotherapy. So it turns out that if the network, the expression associated with the metastasis network was down, the immune activation was up, then if you looked at the survival in that group versus everyone else, then the survival was over 50% versus something like 18% for the people who didn't have this type of dual activation. And so this was something that came about again at the end after I had thought about what had to be done to really properly combine these factors to get an assessment of survival. And the other interesting thing was that factor A does not even, so there's a type of survival analysis that makes certain assumptions called the proportional hazard analysis. Factor A doesn't come out as a good predictor under that. That it was really only by understanding what that proportional hazard assumption meant and looking at the data and looking for a different sort of flag of interest that I knew to put those together with all the other factors and find that in fact when it was combined with immune activation that it was the best, those two were the best combination in terms of predicting survival of any of the other factor combinations. So given this, it turns out I was able to use this information when I recurred in early 2015, which I had a very poor prognosis, and my oncologists were telling me to take more conventional chemotherapy, which I knew hadn't worked, but based upon markers in my tumor from CoreX and some other information from the research literature, I chose to do a different therapy and went into remission sometime in 2015, mid-2015. This is a picture of me and my family, happily then in autumn 2015 after I've gone into remission. And, you know, a little worse for the wear, but still here. And so, you know, the key takeaways, I think this obviously turned out really well from my perspective, and hopefully from Greg's too, is that we're always driven by solving this real-world problem. We were very patient with the process in a way, and I think it was critical that I think in order to make progress in more especially medical applications, there needs to be more people who can understand both the medical application and the data analysis and machine learning. And so what we really need are more fish birds who can swim like fish, squawk like birds, and be bridges in this context. So finally, I just want to have a slide where I acknowledge all the doctors, all my oncologists who actually performed surgeries on me and gave me treatments, and I wouldn't be here without them either. So, thanks.
Shirley, thanks for telling your story, Greg. Thank you very much. We've got 5 minutes for a few questions here. Yes, gentleman on the side there. Thank you.
Hi. Thank you. So very interesting. I got to say, though, my first little bells start to go off because I see you finding pairs of them, and I think of multiple comparisons, and you obviously are a sample size of one. And so I'm curious about validation. Has this moved on to any sort of trial or study where it can be controlled to see if you can identify, say, a drug that matches based on the markers and achieves better results?
Right, so we're working on that. So I was n equals 1. Again, this was retrospective on a database. And so we're working with other people and groups to try and extend it to other cancers and possibly put it into a trial eventually. But it will be an unconventional trial because it's not a situation where you can give the same drug to 100 people, right? It's a situation where you're trying to tailor the treatment for each individual. So, not yet, but it will happen.
Other questions?
Yes?
Yes. Can we wait for a microphone just down here? Thanks.
Sorry, I may have missed this part, but I understood how you saw the gene expression, but how did you come up with the factor A and factor B combination that were the key pair that increased your probability of survival?
Right. So that was interesting. I tried to sort of describe that a bit. The immune factor came from just a straightforward analysis of putting all the factors through a survival analysis and looking at associations.
I see.
And that immune group was particularly strong, but there were other immune groups that were strong too. The immune system was always key, which was information for me, because that's not something people often talk about with respect to ovarian tumors. The factor associated with stemness and metastasis, so that was interesting because it failed the initial single factor survival test. But I knew that I was looking for long-term survival, and I could see that from the curves, okay, you get a little lift in the survival, but there's not a lot happening. So I said, okay, is there something else here? Is there something that biases toward long-term survival that isn't coming out of this test? And I just looked at all the curves, and it turned out that there was one that failed that filter because it actually had a lift only at the end, right? So it came out as poor under that model. But when it's combined, and it turned out that when I looked at the annotations associated with that, something I hadn't looked at before, only then did I see it was associated with stemness. And so it made a lot of sense. Okay, so that you get a selection for stemness that results in faster chemoresistance and an earlier death. So then only then did I combine it with the immune system one and see that in fact it had the best, the highest rate of improvement in outcomes. So, and I didn't use that information though, obviously in my treatment. I actually didn't take, there's no treatment approved in any cancer for, I think, stem cells. So, but the immunotherapy is what I ended up using. So.
Great story. Thank you very much.
Well, thank you for listening.
Okay. Yeah, one final question here, thanks.
Once again, story is fantastic, and I want to talk a little bit more about the challenges of changing paradigms. So you're clearly approaching care from a hybrid perspective, and I know personally a lot of people who have been chronically hospitalized for various treatments, and there's a strong push against going against standard practices in care. How was your experience in experimenting with your own treatment, given the fact that you are qualified to possibly suggest that? How do you see that progressing as maybe doctors start to admit that their education might be limited and should probably lean on more modern approaches in care? And do you see any issues? That's
a big issue. So I can only speak about oncology. I can't speak about other areas. But in the oncology world, and especially in gynecological oncology, the clinical oncologists, they're on board. When I go to them and I say, we want to do this, like, help us, because all these lung cancer people don't talk to us. But lung cancer doesn't use this sort of gene expression profiling either, because they feel they don't need to. But I think it's obvious that if you use only part of the information, you're not going to get the optimal treatments. So I think clinically they're already on board. The research people are kind of wrapped up in their own worlds, but there are a few people trying to bring the two together. And so this is why I said what we really need is more of an explicit effort to bridge that by bringing research people, embedding them with the clinical oncologists. Because when I approach people, I approach researchers now and I say, okay, there's a patient and we want to work with this person to get a genomic profile and see what we can do. They're always eager to help. If they can, if they have the funds, if they have the capability, they want to help. But there's just, they're not connecting. And I think that's partly just a matter of, focused effort and people saying this is what needs to happen. People who have, say, the funding and the authority to say that, well, this is what we're going to put effort into is bringing this together. Not in a sort of abstract way of saying we're going to fund translational research, because that always stays in the research. And if you fund things clinically, it stays on the clinical side. We need to bridge the gap. And so I'm working on that, and other people are as well. So it will happen. It's just a matter of how fast it happens.
Shirley, thanks very much for sharing your story, and best of luck. Great, thanks.